Okay, back to the slide. So if you remember this from our glycolysis discussion, this enzyme is one enzyme that has two domains, the phosphatase domain that we need for gluconeogenesis and the kinase domain that we need for glycolysis. Okay, this reaction right here, so we've got our glycolysis reaction going this way. This reaction can go both ways, right? Or I should really use my equilibrium arrows a little better, but we can get this one going both directions. Okay, hence it's called bifunctional. And we give that enzyme a different name depending on the direction it's catalyzing. We called it PFK2 if we were making the fructose 2,6 bisphosphate. Okay, that's the kinase. Or we called it this fructose bisphosphatase if we were going back. And that's really where we left it as we talked about glycolysis. But now let's talk about how the enzyme knows which domain should be active, right? We need one side or the other to be active, depending if we want glycolysis or gluconeogenesis to move forward. So how does the enzyme know which domain should be active? And it's controlled by blood glucose sensitive hormones. So first, if you have low blood glucose, that's going to trigger your pancreas to release the hormone glucagon. That glucagon is going to activate a protein kinase. It is going to bind to a G receptor protein, or G protein receptor. That's going to activate adenylate cyclase. That is going to catalyze the formation of cyclic AMP, which binds to protein kinase A and activates it. This should sound really familiar, right? When we talked about signaling proteins. So that's what's happening. Your body's producing this glucagon, and it is your primary messenger. It's binding to that G protein receptor, and we see that cascade go on, and now we have activated this kinase. We know kinase transfers a phosphate group. And so the protein kinase phosphorylates the PFK2 domain of the bifunctional enzyme. So let's look at this. There's a lot on here, so just kind of watch slowly as we go. So if blood glucose is low, so I'm going to write that up here. We're going to stimulate or activate this protein kinase A. And it's going to phosphorylate the PFK2 domain. So when PFK2 is active, let me change color here. All right, so when this PFK2 section is active, when active, what do I want to say? Generates fructose 2,6 bisphosphate and speeds glycolysis. Okay, on the other hand, when the phosphatase domain is active, okay, it is going to degrade the fructose 2,6. Okay, it's going to slow glycolysis and speed gluconeogenesis. Neogenesis. So when you look at this, we have a singular polypeptide chain, something like 470 amino acids. Part of it is our kinase domain, part of it is our phosphatase domain, but only one section is active at a time. When your blood glucose is low, it activates this kinase that adds the phosphate. When that phosphate is on there, it deactivates the kinase portion. Okay, so think about what that means. When your blood glucose is low, you don't necessarily want to be pulling more and more glucose out for the glycolysis pathway. You need to build up those glucose levels. So you don't want glycolysis continuing. You want gluconeogenesis going. Okay, but on the other hand, think about your glycolysis series of steps. If you start building up the fructose 6-phosphate, okay, meaning, and let's see, so here was our fructose 6-phosphate. Here's our fructose 1,6-bisphosphate. So if we start building up high levels of this, that would be an indication that probably we're no longer low on glucose. Okay, and so that is going to activate this phosphoprotein phosphatase. It's going to essentially activate this enzyme that pulls that phosphate back off. Okay, and that is going to activate PFK2, which is going to allow glycolysis to move forward. So it's a complicated step because we have several things, right? We've got our hormones that are released when blood glucose is low, activating this kinase that is changing which section of this enzyme that was for that side path is active, that then determines if gluconeogenesis or glycolysis are really going to move forward. Okay, complicated, but a really great example of how hormonal regulation is coupled with this allosteric regulation and covalent modification. All right, let's drop down to this third section. Okay, and we want to focus in right here on this fructose 1,6 bisphosphate. And if you remember, when we talked about glycolysis, that was the product of this step. And we said that it worked as this feed forward effector, this positive effector that says, hey, we got a bunch of this stuff. Let's get that last step going and pull everything through those reactions 3 through 10. We also looked at alanine. We said that if we look at where pyruvate and alanine is here, they are essentially one step apart. Alanine's an amino acid that's one metabolic step from pyruvate. So if this level is going up, alanine, that says, hey, we've got plenty of pyruvate. Let's slow down glycolysis. Okay, now let's look over here at the acetyl-CoA. That is right here. So acetyl-CoA is kind of one large step from pyruvate. Pyruvate's converted to the acetyl-CoA, which then enters the citric acid cycle or gets used for fatty acid storage. If this is piling up, that's an indication that the energy needs of our cell are met, fatty acid synthesis is probably all taken care of. So we don't need to continue those processes, and we can instead start building back up our glycogen stores. Okay, so it is a positive effector. And then we have our energy effectors, right? If we have a bunch of ATP. That's a sign that our energy needs are met, so we can slow glycolysis. On the other hand, if we have a bunch of ADP, that's an indicator that our energy needs aren't met. We really can't be using pyruvate to go back through gluconeogenesis. All right, we need to, that ADP will bind allosterically to each of these and slow them down because we need to be moving our glycolysis portion forward. All right, let's look at a couple questions. So what would be the effect on an organism's ability to produce glucose if there was an inability to produce adequate amounts of pyruvate carboxylase? So let's look at this. Pyruvate carboxylase is right here. So it's that very first enzyme step in gluconeogenesis. So going from pyruvate to the oxaloacetate. And we want to know what possible things would be. So let's think about them step by step. So if we could not make pyruvate carboxylase, would that cause hypoglycemia? And definitely, if the person hasn't eaten recently, this is the very first step in forming glucose from pyruvate. So if we can't do that, we can't bring our blood glucose levels back up. So definitely I would say yes, that one is likely to occur. All right, remember that in the liver, lactic acid can be converted to pyruvate and then gets carried into gluconeogenesis. Okay, or this is a way for lactic acid to be cleared from the blood. And so, but if we can't carry out our gluconeogenesis, we would definitely start seeing this lactic acid build up. All right. If we look at the third one, the body could compensate. So in A, we said yes, hypoglycemia would be really likely. In C, it says, well, you know what? Our body can produce glucose from glycerol. And if you go back and you look at our steps, you'll see that there's an entry point for glycerol. All right, but it turns out that this is a minor pathway. All right, I wouldn't have expected you to know that now or before this, but this is a minor pathway and it's not going to be enough to keep your blood glucose levels constant and prevent hypoglycemia. All right, D, body would compensate by producing glucose from amino acids. That is also true that there is a route to get pyruvate from amino acids that could enter into gluconeogenesis and enter in that oxaloacetate area. It's a route, but you would need to have a huge excess of amino acids for it to be one that your body would readily use, or it would have to break down muscle. So, not major pathway. Again, not something you would have known before this. All right, I would have expected you to be able to predict A and B, but I wouldn't have expected you to predict that C and D would not be strong enough routes to keep your blood glucose levels constant. Okay, so pyruvate carboxylase deficiency is a rare disorder of carbohydrate metabolism. There's several types and they vary in the seriousness of their clinical outcomes, but all of them are characterized to at least some extent by hypoglycemia, okay, and lactic acidosis. All right, let's look at one more question. So the answer to that, by the way, the best answer would be A and B. But I really just wanted to talk through those ideas. All right, what would be the effect on the organism's ability to use glucose as an energy source if a mutation inactivated glucose 6-phosphatase in the liver? So if you remember that very, very last step of gluconeogenesis, we have glucose 6-phosphatase removing that phosphate group to get us glucose. All right, so we need glucose as an energy source. And the first thing is, well, no effect because you can eat glucose. But the key is you can't eat glucose 24 hours a day, right? Short of being hooked up to an IV, you are not going to be able to maintain your glucose blood levels just by eating. You'd have to, and in fact, some people do have to wake up periodically to eat who have deficiencies in this. Okay, we definitely would expect that during the night, because you're fasting during the night, those glucose levels could drop to a dangerously low level.